now let's talk about food preservation. How can we preserve our very favorable food? So to actually understand food preservation, we have to know what are the conditions for the microorganisms to grow. We have talked about them, the environmental conditions, the intrinsic and extrinsic factor, and all these things. Now another thing we must know when you talk about the food preservation techniques, and that is the division and growth of a bacteria. What is the normal way of growth of a bacteria? We have suppose one bacteria. This one bacteria will grow into two bacteria, and these two bacteria, each of those bacteria can grow into another two bacteria. What we've got four bacteria. Now those bacteria will go on to produce more offspring. So we've got 8 bacteria after third round of division, for example. Okay, so that's how bacteria can go. So if you can look at the division of bacteria, it is going on exponentially. So we have an exponential growth of bacteria, right? We have an exponential growth. Of bacteria all the time. It's going on. If we draw a curve of bacterial growth, what we find is a characteristic curve is like that. If a simple load of bacteria, that's why it's not starting from the set point 0, it is coming from this place and it gets stabilized and then gets fall. So this is a normal curve of bacterial growth. If we're talking about the bacterial load in this axis, and we've got time in this axis, so this is time, for example, time, which is a independent variable, and the dependent variable here is a bacterial number or bacterial content. If we talk about the bacterial content in this y-axis and the time in x-axis, what we can find this curve like this. It is called the bacterial growth curve. In this growth curve, we found several different stages of bacterial growth. The first stage of bacterial growth, what we can find here, is this, which is an exponential growth. When the bacteria is dividing like this, which is an exponential growth, 1 to 2, 2 to 4, 4 to 8, like this, and so on. And after some time, it will have a halt place. In this halt region, it will be changing from this rapid growth towards stabilized growth. We call this phase stationary phase. In the first phase, when they grow in exponentially, we call it the exponential phase or log phase. And after the log phase, we have the stationary phase. Why we have the stationary phase? Because after some time, the food material is getting exhausted, the food material is getting lower and lower, and finally they have a survival chance of falling. And some of them are surviving, and no other division is possibly occurred. And not only that, but also the toxic material, the junk part of the bacterial cell is also increasing, because bacteria eat foods, and after eating food, they need to release something in the outer space, because they do not need all those digested junk material in their cell like us. So they need to release them. When they want to release those particles, they are releasing that. So their environment is surrounded by the junk materials or the toxic materials that they secreted after the digestion of the food, but they are also lowering down the food content. And after some time, that's why they reach the stationary phase. And after going through the stationary phase for a long time, they have a rapid decline in their growth phase. Why? Because in those situations they run out of food, and they run out of water content and all these things, and finally all those bacterial population are came to exist or came to be seized. After some time they will fall. So this is a characteristic curve of bacteria. So if we want to prevent the bacterial growth in different places, we can do that. So we need a particular starting culture. So from this actual graph, where we have several things to conclude, we need a particular load of bacteria to be triggered in different environmental situations to grow in food. So we need a normal load of bacteria in food. This is general load or starter load of bacteria. If we do not have a starter load of bacteria in food, so the food, if it is sterile, if we store the sterile food in anaerobic conditions, there will be no chance of food contamination. But normally what happens in these all foods, we can have lots of amount of initial load of bacteria, and this initial load of bacteria helps this bacterial load to take on the whole food. And if we want to prevent the food growth in this exponential phase, it is really difficult, because they are growing really rapidly. We need to eliminate, if we eliminate the food content really rapidly, if we eliminate the water content really rapidly, then we can make this stationary phase come earlier of that time. So after doing that, we can make this after, for example, we have treated this food with different enzymes or different particles that makes this food really dangerous. For example, then we can do is this, the bacterial load will be fall, and we can make this stationary phase come earlier during this bacterial life cycle, and they usually enters into the stationary phase many times before. So this is the time before they can enter it, so we can prevent them by doing this. So knowing the bacterial growth property, knowing the bacterial curve, we can use a different property of different environmental manipulation to work on bacterial cell and to stop their growth in the food. Okay. So commonly what is happen, we can remove. So normally what are the basic techniques for food preservation? There are many techniques. There are maximum actually 3 techniques. What is this? We make the food we make the food in such a place that they never caught microorganisms. That's the first thing. Second thing, we can remove microorganisms. We can remove the initial load of microorganisms which is placed there. The third thing, we can kill the microorganisms that present in that food in those situations. So these are the normal thing. The first thing, which is the removal of microorganisms, that usually be established by the filtration method. There are ultra filters out there, which is for 0.45 micrometer, for example. So you have filters like this micrometer. These filters are really small pore size, like this. The diameter of the pore is 0.45 micrometer. So through this diameter, bacteria cannot pass through. So bacteria can be filtered using these ultra filters. We commonly use this filter for in case of water, beer, wine, different juices and different soft drinks. So all of these other liquids we can use this filter, because in case of solid materials we cannot use the filter technique. We need to have some other technique. So normally food preservation can be done by manipulating different temperatures. So now what we are doing, we are actually killing those microorganisms which are there, and what we are doing, we are actually lowering the load of microorganisms which is there in our food via the manipulation of the environment. Here we have lower temperature. If we refrigerated our food in 5 degrees Celsius, it is not stop microbial growth, but what it does, but microorganisms can still cause spoilage with extended spoilage, but they cannot do the extended spoilage. Growth at temperatures below -10 degrees Celsius has been observed. So that means the food in the refrigerator is not at all safe. So in the 5 degrees Celsius in the food, all those enzymatic activity of the food is halted for some time. It's not that all the enzymes are killed or the bacterial cells particles are killed. Nothing is killed there. So everything is there. Only they are going into a hibernation. When we make out that food into the normal environment, then the food can be spoiled by the bacteria which present there. So that's why it is said that we have to eat a food immediately under 4 hours after taking out from it refrigerator. So if you take out a food from a refrigerator, you have to eat it under 4 hours. If you delay that, if you doing this in case of meat, then do it under 2 hours, because in those hours what we are giving, we are actually giving the rapid environment to grow of those growth of those microorganisms, because for a long time those microorganisms are starved. They are placed in 5 degrees Celsius temperature. So when you take them out, they have found their normal optimum temperature, and when the temperature actually is converted into this normal room temperature like that, then those bacterial enzymes are again activated, and they can help the bacteria to grow and take food particles and make cellular respiration to grow on, and the cell will divide rapidly in those situations. And finally we have a huge load of bacteria. So that's why if we have to eat the food after refrigerator, or taking out it after refrigerator, we have to do it under at least 4 hours. We can use high vacuum canning to prevent the spoilage, because via the canning process we are heating that food about 115 degrees Celsius to 125 degrees Celsius. That's really dangerous. That's really dangerous for us to kill spoilage microbes, but not necessarily all microbes can be killed via this canning process. But if we use this canning process, we have high temperature treatment. Also we have the what you call the anaerobical treatment. In this anaerobical treatment, also some of the anaerobic microbes can grow, but most of the aerobic microorganisms cannot grow in these situations. Okay. Now let's talk about so spoilage. This canned food can also be spoiled, but there are several different techniques. During the canning process may be different faults, that's why they can be spoiled. For the handling purposes, that can be spoiled, and the under processing can cause spoilage. But most of the time this canned food cannot be spoiled, because they can be stored for a long time as it is. And we can pasteurize food for eliminating different microorganisms. What is the pasteurization? Pasteurization is a process of rapid heating and cooling to keep and shake the load of microorganisms, initial load of microorganisms in a food, but it cannot remove or eliminate all the microorganisms. So it is not a sterilization process. This is a process of eliminating some of the bacteria. So in this case, we kill the pathogens and sustainably reduces number of oxygen. So there are a number of different ways of pasteurization nowadays. The normal pasteurization is the 63 degree Celsius temperatures for 15 minutes. It's a normal temper for 15 minutes. It's a normal way of pasteurization purpose. 63 degrees Celsius temperature for 15 minutes. If we do this, we can eliminate most of the microorganisms that can be grown in the food and that can spoil us. So this pasteurization can also be done in case of liquid foods like milk and wine and beer and all these things. But in case of solid food, the pasteurization technique cannot be applied. What we can apply different variations of this pasteurization that can be a high temperature short time pasteurization, and we have a low temperature long time pasteurization, HTST and LTLT. So what is high temperature short time? The name suggests all the temperature is higher. So in this case, the temperature reaches to 120 degrees Celsius. When it reaches to 120 degrees Celsius almost, it kills most of the microorganisms or load of microorganisms that situation, and as a result of it will damage all. But the time for using this 120 degree 130 degrees Celsius temperature really small. For example, 115 degrees Celsius temperature for 1 second or for 2 seconds, that will be the temperature in case of HTST. But in case of LTLT, long temperature long time, low temperature long time, we use normal low temperature for 90 degrees Celsius and 60 degrees Celsius and all this. We use it for 15 to 20 minutes to do that. And we have ultra high temperature pasteurization. In this ultra temperature pasteurization, we used to use 150 and 160 degrees Celsius temperature to kill most of the microorganisms that can be found in food. So this is for 1 second only in this ultra high temperature pasteurization processes. But this pasteurization are also being done, but in this case. But why the shorter heating times are used? Because the shorter heating times result in the improved flavor. If you use a longer heating time, that results in damaging of flavoring. If we heat it very long times, it will damage the compound that occurs for different flavor. Those protein compounds will be damaged, and as a result of it, the flavor of that food will be abolished. That's why we have to use, and nowadays we are using the short temperature pasteurization techniques to do this thing. Okay, now let's talk about different approaches to prevent food spoilage. So we have been talking about all this different manipulating different environment that can stop the spoilage of different bacteria. So for example, this removal of microorganisms, what we have talked about, we can avoid of microbial contamination during the food processing industry, during the handling of the food, and we have to be a little bit more careful about food making or for cooking and all these purposes. We need to rinse our hand really carefully and need to serve this food without any kind of chance of contamination. So we have to keep in mind that removal of microorganisms is the best part of doing all this, because it's the most least expensive and it's a really hygienic one. And now if we have the contamination in food, then you have the lower temperature for refrigerant. For our normal home purposes, we can use this. Then we can use high temperature. We can also use this, which we call the food blanching, and we use this. What we are doing, we are actually cooking foods. By cooking those foods, we are inactivating the microorganisms growth by heating them in higher temperature. Normally when we are cooking food, we are going sometimes over 100 degrees Celsius. So this over 100 degrees Celsius temperature for a long time can kill all those microorganisms that is present. So cooking food is really important. So don't eat any uncooked food, especially those foods which are perishable. Don't eat perishable food uncooked type. Okay. So, and we have the reduced water availability. How can we do a reduced water availability? By use of different salt contents. Use of different salt contents to drive water away from bacterial, so drive water away from food contents. We can use different halophilic environment, but there are some of those bacteria which can really grow in this high sugar and salt content. For example, okay, they are called halophiles. So you use sugar to make a really concentration of sugar content, thus reducing the water activity or availability for the microorganisms to grow. For example, in cherry and all these different kinds of fruit, we can use this high sugar content like that. And we can have the different chemical based preservation. But one thing you should know about this chemical based preservation, that they are also chemicals. What we are using is to protect against ourselves against chemical, we are using chemicals. So this is an oxymoronic type, but these chemicals have to be safe for us, safe for our health. If we do not use a safe chemicals or safety chemical for our food preservation, that those chemicals we use to make us chemical we use to as our friend can be our foe after some time. So we have to use those chemicals which are called generally considered as generally recognized as safe. We call them GRAS. So generally recognized as safe via the different chemical control boards. So if we use those chemicals, that's fine, that's safe. Sometimes there are different organic compounds, the nitrites, the sulfur dioxides, because sulfur dioxide, nitrites, and all these things can interfere with the metabolism of microorganisms, that can prevent those microorganisms to grow in the particular environment. And we can use the physical use of this radiation schemes, ionizing radiation like gamma radiation, and non-ionizing radiation like UV radiation, to kill all the microorganisms that are present in bacterial cell, that are present in food, sorry. So by use of this, we can make this sterile. We can make the food sterile, and but this is not the end of the sterility of the food. So if we make the food sterile, you need to pack those food in such an environment that do not generally contaminate the sterile food again. So that's another important thing to keep in your mind. And we have microbial product based inhibition. So you can use different product, for example addition of substances such as bacteriocins. Bacteriocins are such as some of proteins which can make holes in the bacterial cell, which can make holes, and thus which can kill bacterial cells in minutes. We can use this different bacterial induced materials. So bacteriocins are actually produced by other bacteria cells. I can use these bacteriocins to prevent foods. We can use antibiotic to prevent some of those foods, but normally antibiotics are not used as food preservation, because antibiotics have several bad effects in our health too, adverse effect too. That's why I do not use these other kinds of antibacterial agents, but we can use them for some of those foods to be sterilized, to get rid of these microorganisms. And we can talk about the food preservation. Here are the different chemical based preservation. We have talked about this chemical based preservation. If we want to use chemicals to prevent our chemical spoilage of food, we need to use those chemicals which are generally recognized as safe. So in this type of food, we have normally these are the actually adequate different food preservatives which are used, which is actually recognized as safe. These are benzoic acid. We can use sorbic acid. We can use propionic acid. So we can use this propionic acid for different breads, cakes, some cheeses, and all this. So this bread, cake, cheese, all these fermented products to go against the molds. We can also use sorbic acid or sorbates against these molds. We can use the sorbate and propionates against this kind of molds. Actually, this hard cheese, these syrups, salad, jellies, cakes, breads, and all these things. If we want to go against yeasts and molds, we can use benzoic acid, because the benzoic acid is really strong inhibitor of antimicrobial load, but they can inhibit the growth of yeast and molds, but not the bacteria. Parabens or benzoic acids can be used for different fruits, for example, to prevent the yeast preservation, yeast contamination, because yeast generally contaminate different fruits. In this case, you can prevent this for pickle, apple cider, soft drinks, tomato ketchup, and all these things. And we can use sulfur dioxide, which is a really dangerous for preventing all. So we can use sulfur dioxide for almost eradicating almost every microorganism from a fruit source, like molasses, like wine, lemon, and other food recognized as this. So we can use the sulfur dioxide or sulfides against a whole lot of microorganisms. We can use sodium. We can use, for example, methyl formate, and all these different things, nitrous compounds. But the sulfur dioxide and nitrous compounds are the most versatile of them all. They can kill bacteria as well as yeasts and molds. But benzoic acid can kill yeast, and the sorbic acid and propionic acid kills molds. And we can use radiation. So this is a physical technique to go get rid of bacteria, to make our food sterile. This is the radiation process. We can use this ultraviolet radiation, which is a non-ionizing radiation. We can use this for surfaces of food handling equipments. For example, if we have to sterile the food handling equipment without any bacteria, we can use this ultraviolet radiation, which does not penetrate the food, but it can clean up the surface area of the food. So if the food has the surface area, if you use UV ray, so the UV ray can strike away from the food a surface area that can cleave the food from the outer surface. So cannot they enter, that's why it cannot make the food whole safe. So that's why you need to use another set of radiation. We can use gamma radiation. If you use gamma radiation, that can go through these food barriers and inside of the food, and by doing this it can make our food really safer for us to eat. Okay, so we call them normally Zapatization. So what is Zapatization? It is the use of ionizing radiation, for example the gamma radiation, to extend the shelf life. Normally what we are doing in case of meat, in case of seafoods like sardine, like shellfish, like this, all different types of food, and fruits and vegetables also can be prevented using this Zapatization, which kills microorganisms in moist foods by producing peroxides from water. As we know, water can be converted into peroxide. So peroxide, when it's divided, it will form water. If we do water and oxygen, so if we have water, we release some of the oxygen, the water can be transferred into peroxide if we add oxygen molecule to it. So what this Zapatization is doing in case of this UV this ionizing, that means the gamma radiation. The gamma radiation makes a help or trigger to make a form hydrogen peroxide from the water, using of water. By making this hydrogen peroxide is really toxic material for bacterial cells, and they can be a very dangerous material for bacteria. They can they can prevent the bacterial cells to grow in normal environments. That's why it's really important for us to use this Zapatization. And now we can do against the microbial product based inhibition. We have talked about the bacteriocins, which are the bacterial protein active against related species. So we have some species which secretes the bacteriocin, which can prevent the other species to grow. We can use this because these are naturals. We can use them for different purposes, for natural uses. So they are not that much harmful as the other chemical agents, but we can use them. Some forms pores in plasma membranes, as I have told before, acts as porins, and by doing this it will disturb the osmolarity of bacterial cells. So whenever something makes a hole in bacterial cell, that's why the bacterial cell ions will goes out and down without any without the regulation of a transport system in the bacterial cell. That will leads to formation of different osmolarity changes, and that will kill this bacterial cell in some times, and in a few time later. Okay, the example is a niacin, which is a low acid food of inactivative Clostridium botulinum during the canning process. So the Clostridium botulinum is really dangerous bacteria which called the toxin, which release a toxin which is called the botulinum toxin. This is a potent neurotoxin. So it's a very small percentage of dose of this toxin can kill a whole person. So it's a really devastating toxin which will paralyze our nervous system in minutes. So we can prevent this Clostridium botulinum toxin by using this niacin. If we use this niacin, or sometimes we call it niacin, which is a really low acid, which can be found in really low acidic food, we can use this niacin, which is a bacteriocin. We can use this bacteriocin in low acid fruits. For example, tomato ketchup. We can use it in wines and beers sometimes to prevent the growth of Clostridium botulinum, and to prevent the formation of botulinum toxin in our food. In our canning industries, we can use this niacin.